Hello, hi, good evening, and welcome to the London final of FameLab 2020, brought to you by the Natural History Museum and Cheltenham Festivals. My name's Khalil Thurlaway. I'm a science communicator, if I don't stumble over my words, at the museum, and I'll be your host tonight. FameLab is an international science communication competition designed to find, train, and mentor scientists and engineers to share their enthusiasm with the public. Participants are here to engage and entertain you, armed only with their wits and at most a few props. The result is often an unpredictable, enlightening, and exciting way to feed your curiosity and to find out about some fascinating scientific research that's going on right now. Before we get going, I'd like to thank the folks at FameLab and Cheltenham Festivals for being so supportive throughout the process of preparing this event and then moving it online. I'd also like to thank all my Natural History Museum colleagues behind the scenes, without whom putting on this event and then turning it into a live stream at short notice would have been impossible. So, with the politeness out of the way, let me give you a brief rundown of what to expect. You're about to see 9 young scientists speak for 3 minutes each on a topic of their choosing. They're not allowed any slides, audio, or video content, but they are allowed small handheld props within reason. They'll be judged on the 3 C's of the communication: content, clarity, and that unquantifiable but unmistakable quality of charisma. After each talk, there'll be 2 minutes for the judges to grill the speaker on the aspects of their talk. The winner of tonight's competition will go through to represent our fair city at the UK final, where they'll compete for the chance to represent the UK at the FameLab International Final, which is normally held at the Cheltenham Science Festival, but this year will take place at the Cheltenham Literature Festival in October. Fingers crossed. But who will judge such a storied and momentous competition, I hear you ask? Well, let me introduce your judges for the evening. First up, we have our very own Erica McAllister, Senior Curator of Flies here at the Natural History Museum and a frequent contributor on radio and TV programs, as well as the videos and live shows we produce here at the museum. Hi, Erica.
Hey, how you going?
Hi, good to see you. So, just to fill the audience in a little bit, what's so great about flies?
Everything. You name it, they do it. They're there. They behave in incredibly weird and wonderful ways. They're ignored, much to everyone's detriment. And they're basically giving you all sorts of things you didn't realize. They're getting rid of your dead bodies. And they're giving you things like chocolate. Gotta love the fly.
Chocolate and disposing of dead bodies. Who could want more? So, are
Absolutely.
you going to be looking
The
out
girl's best friend, surely.
or an alibi? So, are you going to be looking out for anything in particular from tonight's contestants?
I like novel facts. I like to be amazed. I like to be able to go away. I don't want to hear anything. You know, you hear similar things again, and people outside of their subject think, or people inside their subject don't think that everyone else reads a lot. So I want to be totally like, oh my God, that is fabulous. That sort of level of like, I love it.
Okay, so you're hoping to be wowed and surprised. Well, hopefully we'll have a good platter on for you later. Okay, I'll catch you later, Erica. Next up,
Good luck, everyone.
Helen Sersky, who is a physicist, oceanographer, author, and broadcaster. So, Renaissance. Hi, Helen.
Hello.
So, what brought you out of the lab and into science communication? Because you were originally a research physicist, right?
I am a research physicist and I'm still a research physicist. I haven't gone anywhere. I've not gone into science communication. I'm just a scientist who talks about what I do.
Sure. So, are you kind of looking for any particular approaches or anything in particular out of tonight?
There's a few things that matter. The first thing is the science has to be at the absolute heart of it. It's not about the presenter. It's about properly sharing a piece of science, a perspective on something that someone might not have come across. And that has to be correct. That's really important. And once you have that, once you have the core of it, then it's about showing it using a perspective that someone might not have thought of. So you must have the robust science, and it's all about the science, but then you put it in a context which makes someone think about it differently.
I guess without the scientific integrity and credibility, you don't have a foundation.
There's no point in being there. If you don't get the science absolutely right, there's no point, really. There's enough people spreading misinformation in the world. We don't need any more of it.
Great. Well, it's great to have your eagle eyes on the team. Thank you so much, Helen. And finally, we have Sam Furness, the Interactive Galleries team leader at our friends, neighbours, and occasionally rivals, the Science Museum. He's also a member of the stand-up science troupe Punk Science. Hey, Sam.
Hi there, Claire. We're all friends. All of those museums, Exhibition Road. It's all lovely.
Exactly, the South Kensington crew.
Yeah.
So, what are some of the joys and challenges of working so closely face to face with the public?
It's that connection with people that really has kept me doing it for so long. Nothing better than seeing a child give you the amazed, like, oh, wow. But even better,
Penny
is when,
dropping moment is beautiful.
yeah. But when the child and like the parents and the whole family do it together, that's exactly what the job is for. And now, kind of managing and kind of when staff start, you can see them when they first have that moment. They kind of get that buy-in. It's really special to just kind of reveal that wonder, I guess, and uncover amazing things.
So I guess that's something you'll be looking out for tonight from today's contestants.
Yeah, completely. I think for me, so much of it is passion for your subject, but also knowing how to give that passion across. I think my biggest advice has always been people like to watch you have fun. People like to watch people have fun and enjoy themselves. So yeah, I mean, Helen's completely correct. Science has got to be there, but also, why do you love that subject? Why should I love that subject? I think that's a very important thing to give over.
Well, hopefully if our contestants tonight can get over their nerves, then they'll be able to show their joy and passion in their speeches. Well, thank you, Sam. Looking forward to hearing from you later. So with the judges in place and the scene set, let's kick things off. For the audience at home, please do feel free to get involved in the YouTube chat. It can be a bit daunting for our contestants to speak to a camera without any audience feedback. So let's give those applause emojis a good workout and keep the vibes going. First up, we have Hanan Alibusetta. Hanan, are you there? Okay, so we're just having a bit of trouble getting hold of our first contestant, but we should have her soon. In the meantime, I hope we're looking forward to it. We've got a whole series of very different speeches lined up for tonight. We've got 6 different speakers from different institutions all around London who all work in different areas of science. So whatever your scientific taste of preference, we should have something for you. We're just still trying to get hold of Hanan. Okay, I think so. We have her. So, Hanan, if you're there.
I am there.
So, when you're ready in your own time, take it away. You have 3 minutes.
Are you going to tell me when I'm nearly there, or?
So we'll put it in. We'll put a notification in the chat so you can see when you've got 1 minute.
Okay, that's perfect. All right. Okay. I don't see anybody else. Right, it's predicted that there will be 27.5 million new cancer cases worldwide each year by 2040. That's a massive increase of about 61.7% from 2018. Therefore, there is an urgent need for the development of novel cancer therapeutics. Now, we can go about it in a way of going traditionally to making a new molecule from scratch, but this might take a long time for them to be developed and out in the clinic. However, we can also repurpose current anti-cancer drug in a different way, but more in kind of more efficient way. How do we do that, and why do we do that? Current anti-cancer drugs are actually very, very potent. They can kill cancer cells, but at the same time, they've got the problem that they actually affect and kill normal cells. Another problem of current chemotherapeutics is resistance. So what happens is after a cycle of chemotherapeutics, the cell can actually develop ways and becomes clever in pumping out those anti-cancer drugs, so the drug actually becomes inefficient. So what we're trying to do in my lab is develop novel ways of delivering those existing anti-cancer therapeutics to target the cells more specifically, cancer cells, but at the same time really overcome the problem of resistance. How are we achieving this? Is by delivering those anti-cancer drugs in a very small nanosite delivery vectors that will mask the drug, so the body and the cancer cell can no longer see it, can no longer see that drug, and will overcome the problem of resistance. But also those really small nanosite delivery vectors can actually deliver the drug more efficiently into the cancer cells. How does it do that? As I said, we can basically, as an example here, you can think about this nanorocket as a nanosite delivery vector, and the astronaut here as my drug. So what you can see here, if you think about this nanorocket as being very, very small, up to the 10 to the minus 9, so in the nanometer range, even smaller than my hair, this basically would imagine that this is so small to navigate inside the body, but it was also very well tailored to reach into cancer cells, but not the normal cells. Now, imagine that this nanorocket can actually navigate the body very well and have the possibility on choosing where it can actually lend into. The cancer patient will definitely then be very happy with this therapy, but at the same time, they can also suffer no side effects like hair loss and stomach problems. Thank you.
Thank you, Hanan.
Thank you.
Judges, do you have any questions for Hanan?
It was very stressful. Sorry.
No, it's good. I have a question. Yeah. Hi, Hanan. My question is, this sounds like a great idea. What makes it hard to do? Why hasn't it been done before?
It's not that it's actually not been done before. I mean, there are some nanosite systems like liposome, which are lipid vesicles that contain anti-cancer drugs. The problem with it is really kind of trying to always shift and avoid the healthy cells. So even those existing nanosite systems that are already in the clinic still have a bit of side effects in there. So the idea is you can actually have, if you can take the rocket as an example, you can actually target targeted ligand. So cancer cell has got particular receptors or overexpression of particular receptors, and you can then target this nanorocket or this nano-sized system to only go into the cancer cells, but not in go to the normal cell. So the thing that you have as well with those nano-sized systems is there are two types of them. So there are things that can be biodegradable and others that are not biodegradable. And those are the problems where you're thinking you're injecting something in the body, and if it stays there for longer, what are the side effects? And that's usually what is limiting the development, I would say, of other nanovectors. Question. Answer your question?
It does, yes.
I got a slight question. You said there was going to be just around a 61% increase from 2018 in 2020. Is there? Well, what do you think the reasons are for that? Is there
Well, the reasons can be multiple. Well, the reasons can be multiple. At the moment, we don't really understand a lot about cancer. So there are certain things that we understand, but there's quite a lot that we still don't understand, whether, for example, the environment, the dietary. So it's a multifactorial disease that different elements, including genetic, can play a role. And again, the change with being in a developed country, things are changing. So there might be a possibility that the air pollution, the food that we eat, what we do, can all kind of be leading to this increased numbers. But at the same time, we don't also have therapies, or we do have therapies, but they're not really, really specific. So it's kind of, we're in a very limited area or space. Let's put it that way.
Well, that's it for our first competitor. Thank you so much, Hanan. Next up, we have Olatz Sachs. When you're ready, if we have Olatz.
Hi, Khalil.
Great. You ready to go?
Yeah, I think, I do think, well, yeah, has to go.
Great. Well, in your own time, go for it.
Yeah.
Your time starts now.
Thank you. So, if a child always forgets a lunchbox and constantly interrupts their teacher, you are going to think that this child has attention deficit hyperactivity disorder, also ADHD. But always forget your keys and constantly interrupt your friends. Could you have ADHD? Well, when I was a kid, I really struggled with reading, and I thought that the other kids were just pretending to read Harry Potter. But I had a very strong super achiever. However, that changed when I became an adult. I really struggled with following the conversation with my friends, and I even got kicked out of the library because I used to stand up too much. So was it because I did not care about my friends? Well, ADHD, impulsiveness, and hyperactivity affect each area of your life, and it impairs it. It's not that it was just very hard to find the chorizo in the supermarket. Actually, in just 4 months. So how come that happened? Did I just metamorphosize, like in Kafka, into a 25 years old ADHD positive? And am I alone? Well, an estimated 1.5 million in the UK, and 92% of them remain undiagnosed. So why is it? Well, some people think that adult ADHD does not exist because it gives a reason for the pharmaceutical companies to keep on producing smart pills. But also, symptoms might just disappear when you're an adult, such as acne or insecurities. And also that could be because some kids might find the adult environment a better environment to tailor their needs in order to remedy the difficulties that make them longer. So if a kid is hyperactive, they might find a job that allows them to move more. But if we follow the same logic, what if we do the other way around? Actually, some people might find that the environment to trigger the ADHD symptoms and consequently have the diagnosis. In conclusion, adult ADHD, it is a real thing. It's a constant battle between and if we don't identify those individuals at risk, we will not be able to support them in their daily activities, such as not keep on forgetting to put the mask on when we go to the shops or keep on listening to me. Thank you.
Thank you, Olatz. Well done. So I know it sounds weird with just me clapping, but now we have all the judges clapping.
My flatmates are clapping. Clap.
So, judges, time for the Inquisition.
Okay, I just there was a little pause in my video just after you said constant battle between. It sounded like a very important sentence, so I just wanted to make sure.
It's a constant battle. Constant battle between nature and nurture, like gene environment interaction.
So what do you think where are the next challenges? What are the next big steps that science can take in order to help with this problem?
I think that, for instance, in women, there is a social. There are a lot of misdiagnosis with as well because of the social expectation that we have. They get diagnosed with anxiety or depression. And then I think that maybe the way that science could do is to actually have different questionnaires that the doctor follow in order to diagnose the person. So if we actually find a way to do the proper diagnosis and also see when that overlaps which other disorders, that would allow us to tackle the anxiety or the symptoms that they might be experiencing. And just this year has been found the first gene for like ADHD. So I think it's I think it is getting there.
I'm really sorry. I'm getting very little of this, so I'm going to have to ask a very broad question. This is something that's very close to your heart, obviously. And how much do you see you being able to add to the science, to add to the research associated with this?
Sorry, no, say it again. It cracked a bit. Sorry, can you repeat?
Yeah, I think you and I are having a really bad link.
Yeah.
If anyone else understood me and was, I'm about you and how this research has impacted on you, and how do you see you being able to move this research subject along?
So how do I find myself researching about ADHD?
How are you involved with moving the question of how we can understand it more?
Team group in my university, and we are trying to actually, so I'm dyslexic as well. So ADHD is more of a spectrum of disorders, and then we are trying to actually help people to recognize this, kind of like tackle the stigma about it does not exist, it is a problem. And also I think that most of adults, they do not, they might not know, they might not recognize it because they have some coping strategies. So I think that a very good way to open it, shamed, and do more talks about it. That's what I'm doing right now. And that's why I'm presenting it here.
Excellent.
Well, thank you so much, Olatz.
Okay.
That was absolutely fascinating. I'm sorry about the connection issues, but I think the judges managed to get the majority of your talk. So next up, we have Varun Ramaswamy.
Hi,
Great,
yep.
Welcome. So in your own time, you have 3 minutes. Go.
Okay. So good evening, everyone. First of all, it's really nice to see you're all safe in the midst of this pandemic. I'm Varun, and I'm doing my PhD in cancer structural biology. Now, I'm sure you all heard of how deadly and unstoppable this disease called cancer is. But if I had to describe it in just a sentence, then I would say cancer is a story about a single cell that just loves to multiply until it occupies every one of our organs and eventually shuts us down. Now, ironically, though, these demented cells that multiply came from perfectly normal ones. And that's exactly why it's difficult for medicines to tell the difference between cancerous cells and non-cancerous cells. Now, I'm working in a team that's trying to identify unique weaknesses within cancer cells that we can exploit to turn the battle around in our favor. Because no matter the similarities with the normal cells, there should be something that makes a cancer cell become cancerous. And I'm happy to announce that I am working on one such unique weakness myself. And this target of mine is a protein that's extremely essential for a cancer cell's functioning. My job is to image this protein under an electron microscope and try to calculate its 3D atomic structure, because we structural biologists believe that if you want to know how something works, you first start by finding out how it looks. And so far, what I've deduced about the shape is that it's quite interesting, to say the least. It literally has a long, thin shaft with 2 round appendages at the bottom. I'm not making this up. And what's more interesting is the way it uses this shape. You see, a cell is filled with a dense network of thread-like filaments that act like a skeleton of the cell. And this protein of mine actually uses its 2 round appendages to hop on these filaments while grabbing hold of it by the tip of its shaft, sort of like this, if you can see me. 1, 2, 3, 1, 2, 3, and so on. Now, at the moment, we have no idea how it's able to do this, and that is a mystery I plan to solve during my PhD. But what we do know is that when it hops about like this, it helps the cancer cell multiply. Now, you're probably wondering, don't normal cells multiply too? Well, they do, but they don't use this protein to do that. It's only when a normal cell's entire machinery turns upside down and it finally becomes cancerous that this protein is summoned to action. So my target protein is unique to cancer cells and is extremely crucial for a cancer cell to work. And that means if I can finish my PhD and get a few answers, we could possibly get one step closer to that medicine that can finally tell the difference between a non-cancerous cell and a cancer cell. So, ladies and gentlemen, in conclusion, I guess this little hopping target of mine can be our very key to hopping away from cancer, quite literally. Thank you for listening.
Fascinating. And I don't think any of us were expecting a shaft-shaped protein with two spherical structures. So I think that's brightened everyone's evening. Judges, have you got any questions?
Yeah, I was wondering how confident you are that this type of protein occurs in all cancers. Obviously, they vary quite a lot, that's obviously one of the difficulties with studying them. So, do you know already that it's present in all of them, or is it just most of them, some of them? How does that work?
So here's the thing: it occurs in a few cancers definitely in a few cancers that have this particular trait in them. But then the thing about this trait is it's really fascinating that it appears in every single cancer at least once along the way of the progress of the disease. So far, I guess in a general sense, I can say it does occur in every cancer, but to what extent it occurs, that is what we have to think about.
Can you tell me a little bit more? You said you're you're scanning them, 3D scanning. Are you doing anything like micro CT tomography? Are you doing all sorts of funky things like that?
Right. No, so I'm not going into the tomography and scanning area. I'm doing more of a transmission electron microscopy.
Okay.
I literally get 2D shadows of the protein, and I try to figure out the shape from the shadow, if that makes sense.
Yeah. Yeah. Interesting.
I just wondered, do you find when you talk to people about your research, are people surprised by how kind of little we seem to know about, cancer, which is, it has, affects so many people? Are people surprised by how much we still have to work out? Or?
Of course, yeah. So it's a little hard to digest how complicated this little cell is, because we think of it as something that we can't see, and how much can it exactly hold in there? But it holds a lot of secrets. And the thing about cancer research is we have been doing it for the last few decades or more, but we always come across different targets with different challenges before we can completely understand what's going on. And that's the biggest hurdle for cancer research. Of course, with all the manpower devoted in the world, we are coming close to getting there, and there are quite a few drugs on trials already. But yeah, it is very surprising, even for me as a scientist, when I try to read about different targets and notice how complicated the cell is.
Well, fascinating. Thank you so much, Varun. Next up, we have Heather White. Heather, have we got you there?
Hi.
Awesome. You ready?
Yeah, I think so, yeah.
Great. So your 3 minutes start whenever you're ready.
Great, thank you. Imagine you're traveling on a train. You delve into your bag or your pocket and you pull out your headphones. You look down and what a surprise, they've become tangled again. Staring down at this kind of tangled, squiggly mess that you can't untangle, you wonder how on earth do they become so tangled every single time? Now imagine a time where you pull out your headphones and they appear in the most beautiful straight line. Sounds nice, right? Unfortunately for us, I don't think this will ever be the case. But there are objects in the world that exist that are this straight. If you stare up at the night sky and look up at Orion's belt, for example, this is thought to be one of these incredibly straight entities. So when we look at our headphones in a tangled, squiggly mess, and we look up at Orion's incredibly straight belt, how do we know just how straight and how squiggly these lines actually are? Well, my PhD attempts to understand just how squiggly actually is a squiggly line. And I'm using this concept to understand both the development and the evolution of mammals by looking at the squiggly lines present in their skulls. So how on earth did these squiggly lines form in the skull of a mammal? To answer this question, we need to understand their development from birth through to adulthood. Did you know that the skull of a baby is made up of lots and lots of bones. When you think about it, this makes total sense, because we all know when you hold a baby, you have to hold it by its head to protect it. And as the baby develops, and this is because essentially the baby's skull is made up of lots of bones that are separated by very large gaps. But then as the baby develops, these bones later join and the gaps are lost. And we no longer have to then hold, by the time you get to an adult, the adult by its head to protect the brain. But the original gap that separated these bones is still visible by means of a squiggly line, and this squiggly line is known as a suture. And this process of the bones joining and forming squiggly lines, known as sutures, is present in skulls across the animal kingdom. So now picture a platypus, a wombat, a giraffe, and an armadillo. Whilst they're all mammals, they're all incredibly diverse. One lays eggs, one has a pouch for its young, and one roams the savannas, and one has its own body armor. So they're incredibly diverse, but these are just 4 out of approximately 5,000 species of mammal. So now picture this diversity across the entire mammal tree of life. It's huge. So how on earth do we know just how diverse, and how do we quantify how diverse these mammals actually are? I've been working on a way to try and quantify just how squiggly is a squiggly line in the skull of a mammal. We can then use this to understand the skull shape variation and how things like diet, habitat, development, evolution, and climate are driving this diversity. So the next time you're on a train and you pull out your headphones, look down and think, just how squiggly is a squiggly line, and what can this actually tell us? Thank you.
Well, I guess we'll all look at our heads and our headphones very differently now. Well done, thank you. The frustration of pulling my headphones out of my bag is going to be somewhat tempered by having a nice little philosophical muse about how straight that line is.
It might be harder with the wireless headphones, so hopefully it still stands for the majority of people.
So Heather, what do we know about how much these lines vary between individuals? Because obviously you were talking about the history of evolution of mammals, but you've got, you must have variation. I'm sure all of us on this call here have different patterns of squiggles. So how much does it vary between individuals? Is it me and my sister, for example, might we have similar squiggles? How close are the links, and how hard is it to go back and compare that to something like a giraffe?
Yeah, so generally you can have different levels of variation. So within an individual skull itself, you have like lots and lots of these squiggly lines known as sutures, and you have variation between each individual suture within the skull for just that individual alone, and that could be impacted by things like if it's kind of playing a more functional role, so if it's one that's more associated with kind of the jaw mechanics, so getting more strain through those kind of processes. And then also you are having variation across different species with if you compare sutures from one species to another, in that specific suture, we do know that there's variation between that. At the moment, we're not really sure what's causing that variation. A lot of things are thought to be related to diet potentially, and also potentially kind of like habitat as well. So think certain animals maybe burrow, and often they maybe use their heads to burrow as well. So that's obviously adding a lot of strain to the skull. So perhaps that's having an impact. And also you have things like deer, for example, that, locking antlers, especially in kind of the autumn time, that's adding additional strain to the skull at that point. So kind of impacting the variation across species from lots and lots of kind of external factors as well.
Thank you.
Thank you.
So do any of the judges have any final questions before we move on?
I have another one, actually, if it's time for another one.
Sure,
How much of this, are you working also with genetics? Because, like with actually molecular genetics, because there's 2 ways you can study evolution, right? There's the physiological route where you look at anatomy and you follow that. And then there's just looking at genetics. Are the 2 things happening in parallel?
So that's a really interesting question. With my project, obviously, generally, you kind of they often don't overlap, and you often have kind of separate teams that work on kind of the anatomical aspects, and a genetic aspect often is the case, and then they kind of maybe would collaborate to pull something together. In my case, I'm mainly looking at kind of the anatomical side of things, but also we are looking into some genetic related things in relation to the sutures, but more in terms of the development of the animal, because we're able to look at that kind of in a laboratory setting where you've got the development of one specific animal, for example. But it's quite hard to look at that in multiple species. And then we're able to do things like, if you knock out a certain gene in an animal, what are you then seeing by knocking out that gene, and how is the development of the suture happening as well? So that's kind of one of the aspects where I'm linking the two together. But on the whole, the genetics of evolution, that's generally studied as like a kind of separate aspect.
Well, thank you so much, Heather. No. Oh, Erica, did you have a question?
Oh, I'm just curious, how do you measure a wiggle?
How do I measure a wiggle? Oh, yeah, of course. So I've been working on basically different ways in which we can do this. And I've been testing kind of 5 different methods. And they're all kind of built on different foundations. So, for example, there's one that's looking at essentially like a length ratio of a line, also one that's using fractal geometry to do this, also Fourier transform methods. So kind of quite a few different avenues to look at the complexity of a line. And essentially what these methods are trying to do is essentially kick out just one number out of the analysis that I'm essentially getting from this. So I can say essentially the higher the number, the more complex the suture is, or the lower the number, the less complex it is. So it's kind of easily interpretable, generally is the idea.
Cool.
Thank you.
So not a piece of string.
Sorry?
So not a piece of string.
Thank you. Well, personally, I love the idea of having a single measure for wiggliness, as if that's a single factor. Awesome. Everything should have a wiggliness factor. So next we have our penultimate talk from Bea Costa Gomez. Bea, have we got you on stream?
Hi, I hope you do. Do you have me on stream?
You're live. We got you. So,
okay, cool.
You ready to roll?
I guess so. It's time now.
3 minutes start now.
Hi everyone. Which is everyone, I guess. I hope people are watching. I'm Bea. I'm a final year PhD student, and I'm an engineer. But I'm a particular set of engineer that has like a more balanced gender quota. I'm a biomedical engineer. And this just means that I work between, the frontier between technology and biology. More specifically, I work between computer science and neuroscience. And this is what I'm going to tell you a little bit about. Because basically, I use the computer to analyze the beautiful microscopy images that the biologists in the group that I work with get. But there are some parallels between what I do and what they do. But before I say that, most of our biology is by studying fruit flies. And you ask, the flies have been mentioned before, but fruit flies are those little tiny ones that are around the fruits, especially around bananas, make everyone go bananas. You ask, why are you using fruit flies? And the truth is that they are cheap. They are very easily genetically manipulate, but most importantly, the ethics committee is not too keen on us going around smashing human brains. So to study brain diseases, we're using fruit flies. And so the parallels between what I do and they do is very simple. They smash the brains of the bugs to get the cells out. I smash my brain cells to get the bugs out. And it works. And we do this to study neurodegenerative diseases. You see, neurons are the cells of our brains, and they are a particular cell that are with us for most of our lifetime. Namely because they don't regenerate and they wire the entirety of our body, and because they have a very specific shape. So if you imagine my arm, and this is a cell body, and these are the dendrites, then you have the axon, which is the long process of the neuron. Now inside the axon, to keep this structure, we find the cytoskeleton, the skeleton of the cell. And there's a particular set of proteins that I'm interested in. They're called microtubules. Now, microtubules normally on the axon look like bundles of dry spaghetti. So just imagine packets of dry spaghetti before the pandemic, and it was gone, but just imagine packets of dry spaghetti all over the arm. And sometimes in some neurodegenerative diseases and also in normal aging, actually, you look and you see the swelling. And then when you look inside the swelling, what you see looks like a bowl of cooked spaghetti, which is fundamentally different from the dry spaghetti. So again, squiggly lines that I'm analyzing, I basically built a software that uses both mathematical methods, but also some machine learning and artificial intelligence to measure just how different the cooked spaghetti is between different diseases. And I could tell you that the diseases are the same or different, but that's not objective. So the whole point of my PhD is to find a way that can quantitatively just separate and tell you whether the spaghetti is the same between the diseases or not. Thank you.
Thanks, Bea. Well,
So
I'm sure.
squiggly lines. I'm sorry, guys. I was so excited. I was like, woo, a different type of squiggly lines. That's good.
Maybe the squiggly line community should get together. I'm sure Erica was into all that fly talk as well.
Yeah, no,
so
definitely.
take it away.
So happy.
Yeah. So tell me, explain more clearly why we use these flies for this sort of research.
Okay,
I know
so
they're big and they're small, but there is.
Okay, so I will give you the Drosophila marketing, as we call it. So basically what happens is that besides there being cheap and easily to genetically manipulate, there's actually about, I think, 75% of human brain diseases can be mapped into genes of the flies. So there's a very high level of conserved mechanisms from the fly to the humans, and they are a much simpler organism to study when we're talking about basic biology, basic mechanisms.
Cool.
How does understanding this help with future therapies? So once you know all this, how does it point the way towards medicine or prevention? How does that work?
So the point is that the point is that these diseases are incredibly complicated, and we don't understand the basics of it. And the whole idea of this is to understand the basics, so that if we understand how it's supposed to work, then we can understand why it's not working, and that's when we go into therapeutics, because we realize the difference in how it actually works.
Well, there's a lot going on. I really enjoyed it, but kind of thinking about AI, that's quite a big buzzword for a lot of people. Do you find when you talk, because I instantly kind of, oh, AI, because you hear it so often, do people get hooked on that aspect? Because obviously it seemed to be quite a small part of what you do. But
So I actually don't do any experimental biology. I do full software, and I built a software which I called Advanced Labeling, Fitting Recognition, and Enhancement of Data, or Alfred. And the whole point is that Alfred does everything on the image processing. So the AI is in 2 steps, actually, of the whole image thing. It goes from processing the image itself, just so we have a clear image of the neuron and we are not actually analyzing, but also in classification. And one of the objectiveness actually is, again, on the squiggly, we're comparing spaghetti, right? That's what we called it. And I can tell you that I think curvature, for example, is an important thing to compare the two, because there's loops. But we're not sure that the parameters that we picked are good. So the machine learning comes in because we just give it blindly. We say, these images are different. Can you tell the difference between them? Rather than asking us questions ourselves. Yeah. Did that tell you a little bit? Did I confuse you more? I'm sorry.
No, yeah. I'm just wondering, is it always spaghetti? Is there any kind of tagliatelle or lasagna or anything like that?
No, it's generally spaghetti.
Generally spaghetti.
And with the name of the software, usually I like to make the spaghetti Alfred. Yeah, that's good.
Well, thank you, Bea, and thank you, Alfred, as well. So for our last talk, we have Shanna Marrinan. Shanna, are you there?
I'm here. Can you hear me?
Yep, we got you.
Hi.
All right, so let's take this home. Last talk. Your time starts whenever you're ready.
Hi. So cannabis, within a short space of time, this may be something which is more likely to be used by your grandmother than by your teenage neighbour. So medical cannabis has received a huge amount of attention lately, both from the scientific community and the public. But is it warranted? And what exactly are the therapeutic applications? And what is the scientific evidence base that we have for this? So cannabis was legalised in the UK for medical use in November 2018, but so far only a handful of people have managed to receive prescriptions for medical cannabis. Cannabis as a plant contains over 400 different compounds. So of course we've got the cannabinoids themselves, but there's also terpenes and flavonoids which give different strains their distinctive profiles. 2 of those cannabinoids have received the majority of the research attention to date, and those are CBD, cannabidiol, and THC, which is tetrahydrocannabidiol. And they have a really interesting relationship. So I like to think of them as siblings. So very different siblings, but within the same family. And CBD is like the sensible older sister, and THC is like the more challenging, younger, but talented one of the pair. And CBD has an ability to mitigate some of the more challenging aspects of THC. So of course, THC is responsible for the psychoactivity within cannabis, which some people find unpleasant if they're using a higher THC formulation. And CBD can mitigate some of that with its antipsychotic properties. They both have very important medical uses of their own. But we believe there is something called an entourage effect, which means that certain cannabinoids may be more effective in combination than they are alone. So what can it be used for? Well, there are currently 4 conditions for which medical cannabis can be prescribed in the UK. And these are chemotherapy related nausea and vomiting, spasticity due to multiple sclerosis, and 2 types of epilepsy, both of which are severe and treatment resistant. However, it's believed that there are over 1 million people in the UK who are currently self-medicating with cannabis for a much broader range of conditions. So research by the United Patients Alliance has found that many people are using it for pain, for arthritis, for fibromyalgia, for ADHD, for autism, for insomnia. And according to the National Academy of Science, there is conclusive or substantial evidence that cannabis can support with some of these conditions, that the efficacy of cannabis-based medicine has been demonstrated for these conditions. And we also know that there's a huge amount of emerging evidence and also anecdotal evidence about its applications for some of the others. So of course, there are side effects, there are contraindications, just as there are with any other pharmaceutical. So people with some types of pre-existing mental health conditions may be advised that medical cannabis might not be for them. We also have to proceed with much more caution when it comes to children and young people, because it may be that especially the higher THC formulations have more of a negative impact on a growing brain than they would on an adult brain. However, when you consider such side effects or such contraindications in light of others with other pharmaceuticals which are used currently to treat those same conditions, it's actually, we can consider it safe. So I hope I've shed some light on medical cannabis for you today. Thank you.
Thank you, Shanna. So, have you got any questions for our final speaker, judges?
I'm interested in how much harder. So,
hi, I can't hear you at all.
Oh, can everyone else hear me?
Yeah, I can hear you.
Oh, you can. I can hear you now. Right.
Okay.
It's just started.
Yeah. How much?
It's gone again.
Oh.
Okay, I can still hear you. Shanna, can you hear me?
I can now, yeah.
You ask a question, Erica.
So I think I can hear Erica. I can't hear Helen.
Can you hear me?
Okay. No, obviously not.
Someone else ask a question.
What's going on?
It wasn't me.
Erica, you have to ask them all.
Can you, I mean, this is obviously quite a contentious subject and one that's going to attract a lot of media interest. How does your how much do you feel that you have to explain properly? How much to get through to people that it's not just a load of people smoking dope? It is actually a very serious and very important piece of research.
Absolutely. I think that has been one of the main barriers, has been public perception and political perception of this. And it is absolutely a pharmaceutical, or it can be many different pharmaceuticals, because, you know, as it contains 400 different compounds, there's lots of different formulations and things which can be achieved from this plant. But yeah, that's absolutely been a challenge. And also the lack of randomized controlled trials, because there's a huge body of data. It isn't always in exactly the right type of package that we like to see, that kind of gold standard within research. But it has been very difficult to do those kind of placebo-controlled trials or comparing it to the next, to the current available best treatment, because it's a scheduled substance, and to do so is incredibly expensive, and there's so many kind of red tape barriers to doing so that until now, I think things are changing. It's starting to get a little bit easier. Hopefully we can start, doing some more of those trials.
Do you two want to ask?
Can you hear me? Hello?
Hello?
I
can't hear anything.
Type in the chat box.
Yeah.
Yeah, maybe if one of you types a question and then either me or Erica reads it out so that Shanna can hear it. Shanna, can you hear me?
Oh, it's just me.
I don't think so. Erica's got that direct line. Okay.
I know. Sam, if it's a quick question, do you want to quickly tell me what it is?
I just wondered if there's countries in the world that are kind of ahead of us on this already, and what countries they are, and are things better there? Are we playing catch up?
Okay, Shanna, he's written, what country is leading in the research, and where are we in the UK in comparison to these other countries?
Oh, that's an interesting question. I think internationally, there's a number of countries which are doing some great work, and of course the Netherlands has been leading in this for some time. But Canada also are quite ahead of us in legislative terms. Portugal has had some innovative approaches, but more again around legislation than research. I think here in the UK, I think it's quite difficult to compare internationally, actually, because one of the other issues is that sometimes, unless data has been collected here in the UK, it isn't taken seriously by our policymakers. So there may be, internationally, lots of data available, but until that's actually picked up and continued here, we're still seeing some barriers.
Cool.
Okay, so thank you so much. I think with the audio issues, we might cut questions there. So that concludes tonight's talks. It's now time for the judges to make their decision. So if you three could retire to the top secret backstage deliberation chamber, enjoy the buffet, and decide the fates of our contestants, you have 15 minutes. That's 1-5, not 5-0. So while the judges deliberate, I was going to fill the time by just sitting here and staring at the camera in silence. But apparently that's not enough and a bit weird. So instead, I've invited my friend and colleague, Cristina Torrente, for a bit of halftime analysis of what we've seen so far. Feel free to send in questions or comments in the chat on YouTube, and we'll try and address a couple during our chat, but no promises. This is our downtime. Cristina, hi.
Hey, Khalil.
Love the lipstick. Looks amazing.
I had to dress up for tonight. It's such a special occasion, isn't it?
Well, I'm really looking forward to having some time to just chat about the science, because I've been spinning all these plates on the live stream. So now we can just relax and have a conversation. So, what was your favorite thing that you heard about tonight? Oh, by the way, we're not going to analyze any of the performances or pass any judgment. We're just looking at the content.
No, that's really
performances.
It's great that I don't have to make that decision, because I really, really enjoyed all the talks. And you asking about the science, it's really tricky. I think they were all really complex topics, and I think that's really, really challenging, especially because they don't have an audience around them. They normally would have at least 50, 100 people looking at them, laughing, and I think it's definitely been very challenging for all the contestants to just get in front of a computer camera and just say the piece. So yeah,
even for professional science communicators like you and me, shifting medium like that at rather short notice can be really throw you off. So for people who are relatively new to science communication, a lot of respect for these guys.
Yeah, I'm just gonna say, for me, they've all done really, really well. Very complex topics, as I said before, which I think is also a challenge. I just loved one moment when they mentioned flies, the fruit flies, and I knew that Erica was just jumping. I was like, she's going to ask a question, and immediately she was like, got a question. So yeah, it's good, but I think that brings it up how the topic not only complex, but they're now in science. I think that you can't just say, I'm just going to study this plant, and what I'm going to do is just take this plant and look at it. You're going to have to apply so many different techniques and knowledge. And so again, you're working with flies, you're working with IA, you are doing, you need to be an expert in so many things to actually do a little bit of science. And you've got a PhD, so you probably know more about this.
Well, I guess it's not just about like one person knowing about a lot of different things, but also increasingly collaborative. You know, nowadays, you would almost never get a research paper with, you know, just one author on it. You'll have, you know, someone who did the proteomics, someone who did the genomics, someone who did, you know, the bioinformatics, and and you end up with these massively collaborative research papers, which I think is a good thing. I think, you know, you get a lot more from having lots of different perspectives on the research.
I've always thought that's really beautiful.
One thing I found really interesting on the subject of kind of different approaches to it was when Shanna was talking about different countries' approach to like cannabinoid research, because it's interesting how a lot of the time science likes to think of itself as kind of pure and objective and existing in a vacuum away from all this messy human stuff like politics and economics and, you know, philosophy and stuff like that. But actually, because science is done by people, it's inherently tied into that whole context.
Yeah, it's social, isn't it? There's not just that hermetic science that happens outside of everything. You have so much to bear in mind. And I think that you talk about Shanna, but even the rest of the topics, you had cancer research over there, ADHD as well, even more complex topics. The moment for example, Heather, who was talking about working with collections, but the moment that she needs to travel somewhere to have a look at a specimen that they don't have in her museum, she'll find herself just having to go through all those different situations internationally. So yeah, definitely. It's just so wide. And I was actually wondering, this was one of the things, I don't have a PhD. So for me, I'm always thinking, just, you know, there must be any other topics that we kind of study on PhDs. And then I watch things like this and I'm like, no, there are loads. Well,
one of the cool things about science is that every time question is answered, it generates all these new questions that you can ask around it. Is that whole thing of when you expand the field of knowledge, you also expand the horizon of ignorance. So it's the gift that keeps on giving. That's why the last bit of every single write-up of any scientific work is, well, if you give me a bit more money, I could answer these questions. Bit more time, bit more money, you'll get these answers.
That's absolutely true. But also the fact that we don't know stuff, and I think that was common from all the contestants. At some point they said, well, we don't know about this, or we don't know about this aspect of this part of my research. And that's really, really interesting. There's always going to be questions, and if there are now, they'll be generated.
Yeah, absolutely. And I think acknowledging that uncertainty as well is really important.
Yeah.
Because if you look around us, you know, you look at the situation we're in now, there's a lot of uncertainty, you know, and, you know, some of the best epidemiologists and virologists and medical ethicists and, you know, social scientists and all sorts of really, really talented, really dedicated people are working really hard to answer a lot of both the scientific and the kind of societal moral questions around this pandemic. But you can only ask those questions by saying we don't know. Whereas in politics, I guess there's an incentive to seem like, oh, don't worry, I know what's going on. I've got everything under control. Don't worry, it's all fine. And I think if you don't embrace that uncertainty, and if you try to claim that you've got all the answers when you definitely don't, I think that can only be harmful.
But I think that's why this kind of events and our work, I mean, not to say anything, but we're a little bit important, like science communicators and scientists that want to go into science communication, scientists that are science communicators. They are so important because, again, we're talking about communicating to the wider public very, very complex topics as the one they were treating in the talks. And you could see them talking about I don't know, wiggly lines, shapes, and flies, and Alfred machines. So I think that's why I'm really, really impressed, because they're taking these complex problems and ideas and just throwing themselves into like translating for all of us who, for us, it will be difficult to understand such complex things.
Yeah, and I think as well, it's especially the charisma part. So, it's content, clarity, charisma. I think the charisma part is really important because you can explain something very clearly over and over again until you're blue in the face. But if you don't make the person you're talking to want to listen, then it's just noise. So I think that, and you and I both know this, that approaching someone's relationship with science can often be more constructive than just kind of trying to fill them up with more science.
Absolutely. And it's just, it just blew my mind that they were so brave to just put themselves out there. And honestly, I'm just glad that I don't have to make the decision on who did best, because they had me like that doing all the talks, and I just wanted to know more. So I think it's going to be very, very difficult for them to decide.
And I hope that either in their own homes off the stream, or once they finish and they can look at the comments on YouTube or something, I hope that they'll get a bit of a sense of how people were appreciating what they were doing and saying. Because just speaking to a webcam without any immediate feedback from the audience can be really, really tough.
It's definitely really tough. But I mean, I think on the other hand, it makes things quite easy for them. I suppose they're not. I mean, I'm on my leggings at the moment. I'm dressing smart, but bottoms are. You don't have to worry. So I suppose. Well, some of them were walking around, weren't they? So they really, really prepared. But I hope that also gave them a little bit of like feeling a little bit calmer in their homes. They can have a drink tucked away over there if they needed it.
I may be wearing a suit, but I'm also wearing Nike Air Max. So.
Nothing against a suit and some trainers.
I guess it's kind of, it's going for a 90s hip hop vibe. If there was Shelto Adidas, then I'd be doing the Run DMC thing.
Khalil, I wanted to ask you something. So I found really interesting that Bea had named her machine Alfred, and that stands for something. It's not like when I call my plants like whatever Trix and Randolph, but when you were studying a PhD, because definitely on my degree I did learn this. Did you guys actually learn how to create acronyms of your projects that actually sound good and make sense? Because honestly, I heard the best things. You read them on papers, you hear scientists talking about it, and I'm like, where did that come from? How did they match everything? How did they get the names right?
That's interesting. Because I myself, I didn't actually coin any new acronyms. I was swimming in a sea of acronyms during my PhD. I was studying immunology. So that's the study of the immune system. And it's very interesting. It's also a lot of very similar acronyms, all in a big jumble with lots of arrows pointing in all different directions, connecting them. And this one promotes this one inhibits this one, but then promotes the other one, but then in certain conditions will do da 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 da. And so I think I kind of had my hands full with this big bucket of immunological acronyms. But sometimes it is amazing how it just like people manage to bring together one. It just works. Especially a name like Alfred. That's just a you got it's like Iron Man has Jarvis.
Yeah, there's plenty. I mean, you've got the classics HAL. Well, it's IBM, but no, I loved Alfred. I was like, legit, like 10 points. Yeah.
Well, we just gotta hope Alfred sticks to helping Bea with her research and doesn't become self-aware and try and take over the world. Yeah,
that's the worry with AIs, right?
I mean, it's a worry in like science fiction, and it is science fiction. It's something that's being discussed in real world like AI ethics, because obviously, the more and more the more stuff we devolve to AI, the more power it has. And that, a lot of AIs, we think of them as kind of these neutral oracles of decision making that are totally devoid of bias. But actually, they're built of bias. They're built by people, and they learn using human data. And so I think we need to be aware that actually we might just be making even more human decisions. And on the subject of that, actually, talking about decisions, it looks like the judges have come to a decision. So thank you so much, Christina. I'll see
Thank
you
you
on
for
the other
having
side.
me.
Cheers. Bye. So I believe the judges have come to a decision. So if we can get them back into the stream, and if they can deliver the verdict. Okay, so drum roll at home, audience at home, drum on your tables.
So do you want the names, or do you want the lead up to it? Because we've got some things to say in general.
Okay, give us the general feedback, and then we'll go with the runner-up, and then we'll go with the winner.
Okay, so I'll go first for the general stuff. That it was great to see so much enthusiasm, this, it's always nice when you have a short period like an hour, and you go somewhere from rockets and cells to mammal evolution via cancer. I mean, that is just brilliant, and it is one of the nice things about these competitions is that you get all this enthusiasm for all these subjects, and there are always more questions that you can ask. So it was great to see so much enthusiasm and good science and interesting storytelling. So those were my good things.
I'd like to say,
Go on, Erica. Go.
I find it very interesting. I like the range. I thought it was very good, and I found that people were very clear, which is really, really nice, especially in subjects I don't necessarily know. It was really good to have such in-depth, such knowledge about their subjects. It was great, and obviously it flies. Well done.
And I just think as well, the weird situation we find ourselves in of streaming everything. It was nice to see that nerves seemed to be a big factor, and everyone seemed pretty calm and confident when they delivered, which was lovely. And great to see, said, a broad range of where science kind of touches everyone's lives, from cancer and then looking to new ways of targeting diseases and some really interesting things. And I'd love to say that I've got I'm full of facts to deliver down the pub at the weekend, but unfortunately I'll just be telling them to people in my living room. I think shout them out the window.
You can deliver them to your fridge or your oven or something. You can talk. You can talk to your can of beer. There you go.
Or you can store them up over time, and then as soon as lockdown is over, you're going to have the most interesting conversation in the pub.
Yes, the best thing is that by then no one's going to care about what anyone says, are they? They're going to be so happy to be in the pub that, frankly, the topic of conversation is going to be by the by. Did you want some winners?
I don't mean to interrupt. I think there might be a bit of an echo on the audio. Does it sound okay to you guys?
Sounds fine to me.
Sounds okay to me.
Okay, might just be my audio. So we've done the generic feedback. Why don't you give us a bit of feedback about the runner-up?
So, shall I from each of us? So the runner-up was, it was so nice to listen because it was such a human. It felt like having a conversation. It felt like a good conversation in the pub with someone who really knew what they were talking about, and that human aspect of it was really nice. And there was a good use of visual analogies.
I really liked it because it crossed over two different disciplines, and the person was able to answer them quite well. Plus, it also brought in, again, with the human thing, it brought in a really recent cultural reference. And I like people being able to compare with analogies to help reference their science. So I was pleased by that.
Yes, and I thought it packed so much in. It did a great job of coming back and wrapping up at the end and kind of putting in a little gift that I kind of understood everything. Because for a while I was a bit, there's so much thinking about here, and then it got delivered in a lovely way.
Wonderful. So this sounds amazing. Who was our runner-up? And this runner-up will be taking the place of the winner if the winner is somehow unable to fulfill their role.
So our runner-up was B with the fruit flies.
Well done, B. Congratulations. Okay, and now time for the big one, the winner. So why don't you give us a bit of flavour to warm us up for the winner?
Well, the great thing about the winner was the clarity of thought, that there was a big idea, we went straight to the heart of it, and then it made it easy to think of interesting questions and to think of interesting things because the way it was expressed was so clear.
The person wasn't bogged down with terminology. They transferred the words from very complicated to very simple that anyone even not in that subject could understand. And I love that ability to be able to communicate like that.
Kept it really simple and kept it tidy, very slick, and could have had the opportunity to get a bit bogged down in maybe some populist kind of humour that would have entertained, but didn't, and kind of just touched around the edge and went for the good science, which was very admirable.
Okay, so who are we talking about?
So our winner with the story of a single cell was Varun.
Congratulations.
Well done. Well done.
We all knew that a subtle penis joke could really go a long way. As long as
Never said the word. It never said the word. I don't think it's.
So I hope we have Varun in the stream so that we can get a few words from him. Not yet? Okay, we're just waiting for him for a little bit. So
Few words from him.
Oh, here he is.
Hi,
Not yet. Okay, we're
Hi,
just
Varun.
waiting for.
Congratulations. You're
Few words from him.
winner of
Here he is.
London 2020.
Not yet. Okay, we're just waiting for.
Right, can I? Oh yeah, you were on mute. I'm sorry about that.
No problem. So have you got a few words for us?
Well, I'm actually ecstatic. And first of all, I just want to say I enjoy listening to everyone, right? From, two ways in which you can talk about squiggly lines in science to cannabis, and especially because it's the 420 month, isn't it? It's April 2020. So I'm sure everyone's thinking about cannabis at least once in this month. I generally enjoyed, and I feel like I learned a lot. And thank you so much. Of course, like you said, Khalil, a subtle joke about whatever can go a long way. I was actually going to try to get a prop or draw it out somewhere, but then I thought, no, it's too vulgar. How do I do this? And then I just realized I can use myself as a prop. So,
and the theatre of the mind can do a lot of work for you.
Yeah.
And I loved your dance. It was a very nice dance. Thank you
so much. Yeah, I was conscious about maybe my head's getting cut off or my toes are getting cut off. So, yeah.
No, thankfully, it did its job.
Okay, that's lovely.
We can only imagine how majestic it would have been on stage. Well, thank you so much, Varun. Massive congratulations to our winner and to our runner-up B. Well done to all of our participants. In fact, you all did an amazing job. Just because you didn't come out on top this time shouldn't stop you from continuing to communicate your science. It's really important and rewarding work, although I am, of course, biased. Before we wrap up, I'd just like to give a massive thank you to all our contestants and judges tonight, to everyone at the Natural History Museum, FameLab and Cheltenham Festivals for making this possible, and to all of you at home for joining us for such a lovely evening. Do check out our website and social media channels for more online content that we're going to keep producing for you during lockdown. I'm Khalil Thurlaway. This has been FameLab from the Natural History Museum. Good night. Bye.